Hello everyone, this is Marzi Khovari. I'm teaching assistant for 2206, which is an online laboratory for as an introduction to plant science. Today I'm going to introduce apple breeding program in the Ohio State University, which is in horticulture and crop science department. Apple breeding program, which is interdisciplinary program in my the lab I'm working, and it's started by Dr. Diana Miller, and then Dr. Jonathan Fresnedo Ramirez has joined to this program. And yeah, I will talk with Jonathan, and he will introduce and explain about this program. Thank you. Okay. Hello, Jonathan. I'm going to ask some questions about your apple building program in your laboratory. And yeah, could you please explain for the students how is it? The first question is that what are the primary objectives of the apple building program at the Ohio State University?
Okay, that is a good question, Marcia, and thank you for your questions. Well, in this case, we are working with what we call short juvenile period apples. These are apples that were genetically modified in order to produce fruit in less than 1 year. The most of the materials that we have in this greenhouse have been already blooming and producing fruit in less than 1 year. And the main goal here is combining novel traits, put them together in new genotypes of apple. What are these new traits? Well, we are talking about characteristics as different flower color, different flesh color, different textures, different shapes of the apples, as well as trying to incorporate some more weird traits that we know they exist in apples, but that they are not currently exploited by the industry. We are trying to understand more about parthenocarpy, what means that we may have fruits that they don't need pollination and that they don't produce seeds. We want to understand how that happens, and trying to understand some other aspects of apple like vigor or production. But the main goal is to make more creative combinations of these traits and have apple trees with all these characteristics, and that we have the molecular markers to follow these characteristics down the road.
Okay, thank you. I'm going to ask a second question, how does the apple breeding program at the Ohio State University incorporate modern genomic technologies into its breeding strategies?
That is a very interesting question. Certainly having this type of breeding program in which our plants that are usually perennial and taking many years to flower is not longer a limitation. We need to introduce some other techniques to making use of those biological characteristics. And one of them is marker-assisted selection or genomics-assisted breeding, in which particularly something that we are doing is genotyping, what means that we are making genomic profiles of each one of these plants using a certain number of polymorphisms that are available in SNP arrays or molecular marker arrays that are not more than chips, in which we are hybridizing the DNA to amplify certain genomic signatures in the apple. That is allowing us to perform different statistical procedures that allow us to predict some characteristics of each plant, like yield, for example, or the content of sugars in the fruit. But also we are using molecular assisted breeding in order to continue tracking some disease resistances, some characteristics of the flesh, or even in some cases the flesh color of our apples, or the color of the skin. Then we continue finding ways and looking for some other options to continue incorporating genomics into the program, because that is what actually helps us to save money in the long term as well as reducing the load of work for people in the labor.
Okay, great. I think you already respond my third question. Okay. Thank you for your time and explanation of your program. Yeah, it was really great. Thank you.
Okay.
Okay. At this part, I'm going to talk to Daniel Hemphill, which is my lab mate, and he's a graduate student, and he's going to talk about his program that he's working on. Thank you.
Hello, Daniel. How are you? Could you introduce yourself for people?
Hello, my name is Daniel Hemphill. I'm a graduate student here at the OERDC under Dr. Jonathan Fresnedo Ramirez and Diane Miller in the apple breeding program.
Okay, sounds good. I want to ask a question. What specific lessons or insight did you gain from your participation in the apple breeding program at the OSU, and how do you envision incorporating this knowledge into your future research or future career path? Thank you.
Thank you. So, I learned a lot of practical stuff in establishing this apple breeding program, or as it is, this accelerated apple breeding program. For instance, we've done PCR. Tomorrow I'm going to be learning ddPCR. I've learned something called an FAMD analysis, which is a factor analysis of mixed data, which is sort of a PCA analysis, a principal component analysis, in which also implements qualitative variables as well as quantitative variables. There's also some more practical things like just the labor required in establishing the program. If you look over there, for instance, we have the seedlings, which are how the apples would look when starting out in the greenhouse. Here are some younger individuals and what the older individuals look like. In between these steps is a lot of practical applications of labor, such as potting, knowing how much soil to order, inventing a fertigation program when there is no SOP for these plants. In case you didn't know, this is an accelerated breeding program where these plants are carrying a transgene that reduces the juvenility period from 4 to 10 years down to 1 or so years. These plants can flower in as early as 6 weeks, as you can see for this crazy individual here.
Nice. Cool.
And there are many other novel things about this program. For instance, the ability to generate parthenocarpic fruits, which here is one I picked earlier.
Oh.
And here's another.
Yeah.
And so, for how I'm going to implement this into my future career, I imagine for how much stuff I've learned about apples, I'll be sticking with them. For how the potential implications of this program, I imagine I'll be working with them for a long time. But all the techniques I'm learning for plant breeding will be applicable no matter with what crop I work with in the future. That's about all I got. Thank you.
Thank you.